all of you, you know, the blood is classified as which type of tissues? Yes, special type of connective tissue because it is in a fluid state. And why we classify it as connective tissues? Because it connects the different parts of the body with each other. It's made up of fluid part, if you call it plasma, and cellular part. And the cellular part made up of three main types of cells, which is the RBCs, white blood cells, and platelets. The blood represents about 8% of the total body weight in a very, very significant way. And it is of 5.5 liters volume in males and less than that in females. And all of you know why the blood is high in males than females. مش كده؟ Why blood volume is greater in males than females?
Is they're bigger. Bigger body.
Well, but there is females are very bigger than me multiple times, yes?
Maria. Menstrual cycle.
Now, menstrual cycle, the blood loss during menstrual cycle is compensated. Immediately we're going to be compensated so as to reset the blood volume for the females. And it is about, normally it is not more than 80 milliliters, very small volume. Right? Yes? That is because of the lean body mass. The lean body mass is the body mass without fat. In males, it's greater than females. The muscles in males is greater than the females. All of you know that the muscles are more active metabolically than the adipose tissues. مش كده؟ It needs more oxygen and it needs more blood so as to transport this oxygen. That's why the female's RBC's number, blood volume is less than the male. As we mentioned, it's made up of two parts, fluid part and cellular part. The fluid part represents about 55% of the total volume of any volume of blood, about 55% of which is plasma, and the cellular part represents about 45, and it is of pH range between 4.5 plus minus 0.02. I prefer this. Here is another reference, but other figures, but I prefer the very narrow range of pH, which is 4.5 plus minus 0.02. Very narrow range because the change in pH is very serious. The functions of the blood, all of you are aware about the functions of the blood, as you mentioned at the beginning of the session, that it acts as a connective tissue to connect different parts of the body with each other, to transport nutrients and gases and waste products, and the mediators which is produced by the cells, target cells, and circulating to their target cells. And they are circulating so as to reach their target cells far away from the site of production. And the blood acts as a mediator for this process, interconnection between the cells secreting the hormone or mediator and the target receptors of the hormone. That is highly important transport agent for the nutrient and gases and the waste products from the side of production to the side of elimination. Also, it plays a role in regulation of pH because it is containing, what is the factor lead to? Containing the bicarbonate act as? Yes, because it's containing buffers. All of the buffers are present in the blood by different concentrations. And the buffers are the first line against any change in pH. They are acting within fraction of a second up to seconds, so as to maintain the pH. The blood containing from the cellular part of the blood, there is cells known as white blood cells play essential role in the immune response. They are acting against invasion of the body with the foreign bodies. And also it contains proteins which act as antibodies against the foreign bodies. The blood containing substances act against blood loss if a cutting of major artery or any capillary is okay. They are leading to healing, fast healing or fast blockage of the side which is injured, so as to avoid excessive blood loss. Because all of you know excessive blood loss will lead to shock. And what is the consequence which lead to the shock?
Death.
Shock is the end, yes.
Hypovolemia.
Lead to hypovolemia. Hypovolemia lead to drop in the blood pressure, مش كده? And it will lead at the end will lead to hypovolemic shock. Hypovolemic shock. Yes. The blood, as we mentioned, made up of fluid part and cellular parts. The cellular part made up of three types of cells. Erythrocytes, which is the red blood cells. The leukocytes, which is the white blood cells. And the thrombocytes, which is the platelets. The red blood cells, all of you know the main function of the red blood cells is to transport oxygen and carbon dioxide. And they are playing a role also in the as buffer because they are containing hemoglobin. Hemoglobin also acts as a buffer. Besides, it is the main molecule which is transporting the oxygen and carbon dioxide. The white blood cells, as we mentioned, play an essential role in the, or crucial role in the immune response, and the platelets are the part of the blood cells which is responsible for blockage of the break or injured blood vessels to avoid blood loss. If we apply a centrifugal force to a column of blood collected, for example, in a tube, if we collected blood in a tube and we apply the centrifugal force to this tube, you know the centrifugal force, مش كده? It acts to the force, it's a rotating force, act to repulse the particles from the center of the force according to their density. هو الطرد المركزي مش كده؟ It repulses the particles according to their density. So when we apply centrifugal force to a column of blood in a tube, the blood is going to be separated into two parts. Fluid part at the top and the bottom part which is containing the cells according to their density. The more dense cells is the red blood cells. It will be pushed towards the bottom part of the tube, to the blind part of the tube, and at the top of the column of the blood of red blood cells, there is a creamy-like layer known as a buffer coat. This buffer coat containing the white blood cells and platelets. If they ask you what is the buffer coat is a layer, this layer, between the column of the fluid and the red blood cells. This layer known as buffer coat, containing the white blood cells and the platelets. And as far as you see, most of the cellular column is red. That is because the number of red blood cells in 1 cubic millimeter is in millions. It's around 5 billion, average of 5 billion cells per cubic millimeter. At the same time, the number of white blood cells in the same cubic millimeter is about around 7,000 cells per cubic millimeter. And the average platelets number is about 200,000 cells. If we compare these numbers of the red blood cells to the platelets and white blood cells, we obtain that there is very, very significant difference between the number of these cells. For this reason, when we packed the cells, most of the column will be of red blood cells. The top part, which is the fluid part, which we call it plasma, it's made up of about 90% of which is water and 10% is solid substances which dissolve in the plasma, which is the gases and plasma proteins and electrolytes and the other molecules which is present in the plasma. The plasma, as we mentioned, represents normally about 55%, and the cellular part represents about 45%. please يا شباب come over this. We are going to talk about all of the content of this table, but after this session, please come over it very carefully. لأنه physiologists like to ask from this table. طيب. As we mentioned, if we apply centrifugal force to a column of blood in a tube, we will obtain two parts, bottom part which is containing cells and top part which is containing fluid part. The fluid part, we call it plasma or serum, according to the tube which we used to collect the blood. If the tube which we used to collect the blood containing anticoagulant, and all of you know what is the meaning of anticoagulant, مش كده? It's a substance which prevents occurring of clot, مش كده? It breaks the cascade of the reaction which lead to a clot of the blood. For the fluid part which we obtain, when we use the anticoagulant, we call it plasma. And also if we apply centrifugal force to a blood column in a tube containing no anticoagulant, also it will be separated into two columns. Bottom part containing cells and top part containing fluid. But the fluid at that time, we call it serum. The fluid, we call it serum. What is the difference between serum and plasma? Coagulation factor. What about coagulation factor? Yes, you are right. But what about it? Where? Is a serum containing no clotting factors. And no, importantly, no fibrinogen. Because the cascade of the reactions which use the clotting factors, the end product of which is to convert the fibrinogen, which is soluble protein, into fibrin, which is solid mesh protein. And this will be tracked with the cells in the column in which we use no anticoagulant. You have the top part containing no anticoagulant consumed, as your colleague mentioned, and also no fibrinogen because it's consumed so as to make the clot. You have the difference between a plasma, where in the plasma, the column at which we used the anticoagulant, no consumption of the anticoagulant is okay, and no conversion of fibrinogen into fibrin. The fibrinogen remains as it is. They get contain clotting factors and containing fibrinogen. That is the serum, the plasma. And the serum containing no anticoagulant and no fibrinogen. And if they ask you where is the fibrinogen gone, it's actually it is converted into fibrin, which is solid-ish. Gene means inactive, مش كده؟ Fibrinogen, angiotensinogen, all of gene here means inactive. When we remove gene, it will be activated. Fibrinogen is inactive soluble protein, and when the cascade of reaction use the anticoagulant, it will be converted into fibrin, which is solid mesh. The plasma proteins are a group of proteins present in the fluid part. As we mentioned, the fluid part, we call it plasma when we use the anticoagulant. These are the plasma proteins. There is a huge number of plasma proteins. But the three types of plasma proteins which make significant according to their concentration are albumin, globulin, and fibrinogen. Albumin, globulin, and fibrinogen. And generally, the plasma proteins play a role in transport of the non-soluble substances and play a role in the mechanism of coagulation. They are acting as transporters and play a role also in creation of the oncotic pressure. All of you know the oncotic pressure, also known as colloid osmotic pressure. The oncotic pressure is an important pressure which regulates the transport of fluid across the capillary membrane. مش كده؟ You know starting forces? عارفوا الإديمة؟ ها؟ مش خاطر الإديمة؟ swelling سببه شنو؟ سببه إنه one of the causes a disturbance in the balance or equilibrium between the fluid which is transported from the intravascular space to the interstitial space and the fluid which is passed in the reverse direction. All of you know the hydrostatic pressure is mainly push the fluid from the intravascular space to the interstitial space so as to supply the cells with nutrient and gas and oxygen. And the oncotic pressure, which created by the protein which is present in the plasma, absorbs the fluid which is pushed by the hydrostatic pressure to return it back to the intravascular space. But it is absorbable pressure, act to absorb the fluid from the interstitial space to wash the waste product and so also to supply to transport the products of the cells with the blood. So if the plasma proteins is decreased, the force of absorption will decrease. What is the factor create this force? Is the particles of plasma proteins. If any decrease in the plasma proteins will decrease the oncotic pressure or the colloid osmotic pressure, which absorb the fluid. Highly important pressure, and there is forces known as the Starling forces which control the transport of fluid across the capillary membrane, of which the oncotic pressure, which created by the albumin, is important. Albumin is the main protein which creates the oncotic pressure. The main function of albumin is to create the oncotic pressure. Any decrease in the plasma protein, or small molecular weight plasma protein, by the way, that's a low, easy loss as the renal membrane of filtration if it is defective. It will lead to systemic edema. If they ask you about the main function of albumin is the main protein which creates the oncotic pressure. The oncotic pressure, highly important pressure, which control the fluid transport across the capillary membranes. Globulins act as transporters, mainly act as transporters. And now why we are transporting substances with proteins? For two reasons, two main reasons. There's other reasons, but there's two main reasons. Why we are transporting substances with the plasma proteins?
Water insoluble.
Now?
Water insoluble.
Water insoluble substances and small molecular weight substances. For example, in the second session of endocrine system, they said to you that the thyroid hormone made up of amino acid and iodine. مش كده؟ Only one amino acid tyrosine with 4 or 3 iodine. Very small molecular weight. But it is so easy to be washed out with the urine when the fluid filtered at the level of the glomerular membrane. So to avoid filtration of this small molecular weight, important hormone will be transported binding to protein, because the proteins normally are not filtered across the glomerular capillary membrane. The reason of transport of substance, the two main reasons and other reasons, either to be water insoluble, as your colleague mentioned, or to be of small molecular weight to keep it circulating in the blood, for a long period of time. And globulins of 3 types: alpha, beta, and gamma. Alpha and beta, they are acting as transporters. Gamma globulins are the antibodies produced by the lymphocytes. And by the way, all of the plasma proteins, albumin, alpha globulin, beta globulin, and fibrinogen, and the other trace plasma proteins are produced by the liver. Only the globulins of gamma region, which is the antibodies produced by the lymphocytes. Okay, Shabab? Well, fibrinogen, as we mentioned, is an important protein in the clotting mechanism. It's going to be converted into fibrin. Here we have 2 terms. Put these 2 terms in your mind well. The first one is hematopoiesis or hemopoiesis, the mechanism of blood formation. And the second one is hematopoiesis or packed cell volume. The packed cell volume is the column of cells out of the whole volume of blood. The percentage of cells at the bottom out of the whole volume of the blood. This we call it packed cell volume because we pack it like this so as to be, according to the density, to be at the bottom part of the tube. But packed cell volume is very important. It will give you a good idea about the state of the subject in relation to the hemoglobin concentration. Because the cells containing hemoglobin, if the number of cells is decreased, definitely the concentration of hemoglobin will decrease. And even if the number of cells is normal and any mechanism lead to rupture of the cells and the hemoglobin is splashed in the plasma, hemoglobin in the plasma is not capable for transport of oxygen. Only the hemoglobin which is responsible for transport of oxygen is that one which is present within the cells. If any decrease in the cells will decrease to the oxygen carrying capacity of the blood, a condition which is known as anemia. مش كده؟ طيب, the blood formation actually started as a zero time of the embryonic life, but it will be detected after 3 weeks in the yolk sac. انتوا عارفين كلكم اليولك sac مش كده؟ One of the membranes which is going to be a part of which is going to be converted into stomach مش كده؟ small intestine to large intestine, most of which the precursor cells started at the level of the yolk sac. ففي week 3 it will be detected the formation of blood cells at the yolk sac. That is in the first gestation, first terms of pregnancy are 3 terms. The first term of 3 months, second term of second 3 months, the third one of 3 months. So we call it the first trimester, second trimester, and third trimester. The first trimester will start at the level of the yolk sac, and then the liver and spleen and lymphoid lymph nodes will take over also. But in the second trimester, the liver will start to be a primary site of formation of blood cells, and then as the third trimester, the bone marrow and the liver are the 2 sites of formation of the blood. After birth, the formation from the liver will be switched off, and the bone marrow, red bone marrow remain the main site of blood formation. All of the bones of the neonates are responsible for formation of the blood cells. And the size of the bone marrow or of the bones, which is responsible for production of blood cells, will decrease by aging. Will decrease by aging. Why decrease by aging? Why decrease by aging? Yes, حبيبي نعم.
Because cells slowly die.
Huh?
Because the cells slowly die and they can't be protein anymore.
What? By aging. Huh?
The red bone marrow changes to yellow.
Right. Oh, yes, very good. Why change the yellow? Very good. Because the red bone marrow mass will decrease by aging. It will be converted into yellow and calcified, مش كده؟ For solidification of bones. The calcium يحصل فيه deposition في bones في yellow, and then it become very solid bones so as to support the body. The red bone marrow decrease by aging, and the number of red blood cells and the volume of the blood in the, please try to open this. The volume of blood decrease by aging also. In early childhood, the neonates, a number of RBCs is very high compared to the adults. Why? Because of the time of rapid growth, مش كده? We are in need for more transporters of oxygen. We are in need for more transporters of oxygen. The red bone marrow is the main site of formation as far as we mentioned, and decrease by aging for conversion of the bones into yellow marrow and calcification of the bones. As far as you see here, the size of the bone marrow will decrease in any bone by aging. And for diagnosis of a group of pathological disorders, we are need to collect a sample from the bone marrow itself. This we call it bone marrow biopsy or bone marrow aspiration. The best site to collect this sample is at the iliac crest. The iliac crest is a more safe site to collect this. Also, we can collect it from the sternum. From which marrow will be obtained from the sternum than the iliac crest. The RBCs are unique cells. Disc biconcave containing non-regular cells, anucleated. The mature RBCs, which is responsible for transport of oxygen, containing no nucleus. And it is disc biconcave. This disc biconcave increases the surface area so as to facilitate diffusion of oxygen to bind with hemoglobin within the RBCs. And this surface area not to increase the surface for loading of the RBCs زي ما شفت في كانيا مكان الحياة مش كده بيجيبوا ال RBCs فيها البابلز of oxygen loading لكن it is not like this طبعا بس عشان بتكون دايما الناس في أعمار صغيرة بيحاولوا to explain it like this لكن actually this flat structure lead to increase in surface area to facilitate a simple interaction between oxygen in the plasma with the hemoglobin which is within the RBCs. And also this flat structure, this biconcave structure facilitates squeezing of the RBCs through the narrow capillaries. It can pass through a capillaries of less diameter than the RBC's diameter itself. Normal diameter of RBCs are about 8 micron. So it can pass through capillaries of diameter less than 8 micron. That is due to this biconcave structure. Increased number of RBCs. Actually, in the blood, there is equilibrium between the production and destruction of the RBCs. A lifespan of the RBCs is about 120 days. After this 120 days, it will be captured by the reticular endothelial system, of which the spleen is important. But there is an equilibrium between the amount of RBC of the RBC is going to be destroyed as a level of the reticular endothelial system and produced by the bone. And this will keep the number of RBCs in the blood a little bit constant. So if there is disturbance between the production and destruction, it will lead to either anemia or polycythemia. Polycythemia increases number of RBCs greater than the upper limit of the normal significantly. And either to be pathological or physiological. Physiological, one subject traveling to the high altitude as compensatory mechanism, the number of RBCs will increase so as to increase transporters for an area of low oxygenation. That's physiological, we call it physiological polycythemia. For increased number of RBCs more than the upper limit of the normal significantly, we call it polycythemia. If the number of RBCs decrease below the lower limit of the normal significantly, we call it anemia. And this will lead to a decrease in oxygen carrying capacity. For this reason, the more accurate definition of anemia is a decrease in oxygen carrying capacity due to decreased hemoglobin concentration. Hemoglobin concentration decreased due to decreased number of RBCs production or very massive destruction. If there is normal production and massive destruction, this will lead to a drop in the number of RBCs. If there is a failure of production and normal destruction, also will lead to a decrease in the number of RBCs. As we mentioned, the number of RBCs in the neonates is greater than the males, and the males number is greater than females due to the normal physiological requirement of the females to oxygen. The transporter of oxygen is hemoglobin. This hemoglobin is a molecule made up of two parts, polypeptide and heme part. The heme actually is 4 pyrolyl rings containing iron. 4 pyrolyl rings containing iron. each protein part, which is globin, by the way, globin, not globulin. Globulin is plasmaprotein, and globin is a hemoglobin protein of two chains, two alpha and two beta, each one of these containing heme, and each heme made up of 4 pyrrole rings containing iron. And actually, the oxygen transported binding to the iron within the heme group. يبقى each molecule capable for transport of 4 oxygen, مش كده؟ They said that each cell containing about 250 million hemoglobin molecule, يبقى each one capable for transport of billion of oxygens. لأن أي واحد فيهم 4 multiplied by 250. The hemopiesis, as we mentioned, is a process of blood formation. We state the locations of formation in the intrauterine life and after birth. And all of the cells, RBCs, white blood cells, platelets, their mother cell is hematopoietic stem cell. Hematopoietic stem cell. Presence of certain mediator lead to differentiation of this hematopoietic stem cell toward RBCs. Other mediators differentiate the same cell into platelets, and outside mediator differentiate this hematopoietic cell into white blood cell. يبقى الميدياتر هو is the factor which lead to differentiation of the hematopoietic stem cell, which is also known as hemocytoblast, into RBCs or white blood cells or platelets. The formation of RBCs, we call it erythropoiesis. Erythropoiesis, formation of RBCs, thrombopoiesis, formation of platelets, and leukopoiesis, formation of white blood cells. Formation of RBCs at the level of the bone marrow, it takes about 7 days. يعني from the triggering time, the mediator trigger differentiation of the stem cell toward RBCs, the sequences of development and division will take about 7 days within the bone marrow. The stage which produced by the bone marrow to the blood or squeezed from the bone marrow to the bloodstream is immature stage, which we call it the reticulocyte. A reticulocyte is an immature last stage within the bone marrow containing remnant of the nucleus. It takes about 2 days at which this remnant of the nucleus is going to be washed out from the cell, and it becomes this biconcave containing no nucleus and even containing no remnant of nucleus, and at that time it will be capable for transport of oxygen. The mediator which is differentiate the RBCs, the hematopoietic stem cell toward RBCs, is known as erythropoietin, hormone produced mainly by the kidney cells, known as erythropoietin. But these are the stages from the hematopoietic stem cell and myeloid stem cell and erythroblast, then reticulocyte. All of these stages are within the bone marrow. Then, as the bloodstream, the reticulocyte become mature within 2 to 3 days, containing no remnant of nucleus and containing no nucleus. Okay? But these are the sequences. مش كده لو if they ask you about the sequences of development of the RBCs, it is pluripotent stem cell, myeloid stem cell, erythroblast, and reticulocyte, and then RBCs. Okay, شباب? طيب, we said that the mediator which is differentiates the stem cell into RBCs is erythropoietin. About 85% of erythropoietin produced by the kidney cells, and the rest 15 produced by the liver and other cells in our body. But this 15 is not enough to maintain a normal number of RBCs. We are in need for this 85% from the kidney so as to maintain the normal number of RBCs in the blood. Any renal failure is associated with decreased number of RBCs, which lead to anemia. Because the erythropoietin produced by the kidney cells is significant for formation of RBCs. What is the factor that stimulates the kidney so as to release this erythropoietin is the hypoxia. Hypoxia may occur due to decreased number of RBCs passing to the kidney, مش كده؟ It will lead to a decrease in the oxygen carrying capacity. Hypoxia, then the kidney secrete erythropoietin. The target cells of erythropoietin as the level of the bone marrow. It triggers formation of RBCs, which takes about 5 to 7 days, and then the number of RBCs will be reset in the bloodstream, and the blood passing to the kidneys with no decrease in the oxygen carrying capacity, no hypoxia, erythropoietin production will be switched off. So this negative feedback we can see. But also, narrowing of the renal artery will lead to a decreased blood flow to the kidneys, مش كده? Also will lead to trigger secretion of erythropoietin, and number of RBCs will increase. This will lead to secondary polycythemia due to narrowing of the renal artery. Okay, شباب? These are the steps of erythropoietin stimulation. If they ask you what is the stimulatory factor of erythropoietin, is tissue hypoxia. Tissue hypoxia. Target cells of erythropoietin at the level of the bone marrow. An outcome of erythropoietin production will be prominent not less than 5 days after the time of stimulation. مثلا حد يحصل له cutting of major artery now, الفاست مانجمنت للشخص ده شنو؟ لو حصل لواحد كار اكسيدنت وحصل له كاتنج لميجر ارتري. انا بس الاسئله دي لانه كنت طبعا
bleeding يعني.
نعم؟
Stop the bleeding يعني.
Stop the bleeding and then
blood transfusion.
yes, blood transfusion so as to reset the oxygen carrying capacity. لانه حصل له loss of excessive blood loss containing huge number of RBCs, decrease the oxygen carrying capacity, decrease the oxygen to the brain. To act against, safeguard against the destruction of the RBCs of the brain cells, which occur if the brain not receiving blood force for 4 minutes, it will lead to irreversible brain damage. For this reason, the immediate management is a blood transfusion. Why blood transfusion? If we discover someone suffering from anemia due to decreased supplementation with iron, which is we are going to talk about it. واحدة من common anemias. And if it is severe anemia, and management also will be blood transfusion. And then تدي الشخص ده iron. لكن الايرون output بتاعه حيكون شنو؟ 7 days after the time of discovering of the anemia. لكن blood transfusion it will reset the oxygen carrying capacity immediately. For normal formation of blood cells, we are in need for nutritional factors, normal nutritional factors, normal iron, normal amino acid, normal any nutrient which is involved in building of the cell membrane. And we are in need for maturation factor. Because for formation of the RBCs, the stem cell will be exposed to the mediator, and this mediator will lead to development and division of the cell. An excessive division of the cell needs division of the replication of DNA and division of this DNA. So for normal replication of DNA, we are in need for maturation factor, which is vitamin B12 and folic acid. Vitamin B12 and folic acid are highly important developmental factors, which is important for maturation. And maturation, it needs formation of DNA. Also, we are in need for our control factor, which is erythropoietin, to trigger differentiation of the hematopoietic stem cell. And we are in need for a normal, healthy bone marrow. Healthy bone marrow. These are the main requirements for normal blood formation, blood cell formation. Anemia, as we mentioned, is a decreased, number of oxygen carrying capacity lead to due to decrease in the hemoglobin concentration below the lower limit of the normal for age and sex. لأنه hemoglobin concentration of the adult is anemia for the newborns, مش كده؟ لأنه as we mentioned, the number of RBCs in the newborns is higher. And also, a geographic area play a role in definition of anemia. A subject from Abha, for example, is polycythemic compared to the number of RBCs for those who are living at the sea level. But at the high altitude, it is normal number of RBCs and normal hemoglobin concentration. It's not a polycythemic. As example of anemias, the first one is the nutrition anemia, and the main common nutrition anemia is iron deficiency anemia. Iron is highly important for synthesis of normal heme, مش كده؟ Heme is essential part of the molecule responsible for transport of oxygen, which is hemoglobin. And there is anemia known as pernicious anemia, and this pernicious anemia is due to deficient or failure of absorption of vitamin B12. احنا قلنا vitamin B12 is highly important maturation factor. Normal supplementation with vitamin B12 and failure of absorption of this vitamin B12, this will lead to this anemia, which is known as pernicious anemia. Why vitamin B12 absorption may occur in some patient? That is due to absence of important factor responsible for transport or facilitate absorption of vitamin B12 produced by the stomach cells, known as intrinsic factor. As a terminal part of the small intestine, vitamin B12 absorption will occur. But when it is coupled with, with intrinsic factor, an absorption will occur to intrinsic factor with vitamin B12. If there is no intrinsic factor, no absorption of vitamin B12 will occur. This we call it pernicious anemia. There is also anemia due to a decreased supplementation with vitamin B12. But it is, at this time, we call it vitamin B12 deficiency anemia, and it is not a pernicious type of vitamin B12 deficiency anemia. The ablastic anemia, ablastic, that means less blast cells, failure of the bone marrow to produce cells. As we mentioned, there is an equilibrium between production and destruction. If production is decreased and destruction is normal, this will lead to a decrease in the number of RBCs. And failure of bone marrow to produce cells may occur due to exposure of the bone marrow to radiation or certain chemicals. It will lead to this type of anemia, which is known as ablastic anemia. Also, there is a renal anemia. As we mentioned, most of erythropoietin is produced by the kidney cells. A decrease in the erythropoietin production will lead to anemia. عشان كده اي شخص تلقى عنده renal failure تلقاه احنا عاملين له supplementation ب erythropoietin, recombinant erythropoietin. And the last one of the anemias is the hemolytic anemias. Anemia due to a massive destruction of the RBCs. Massive destruction of RBCs may be due to a parasitic infection or due to congenital abnormalities. Congenital abnormalities in the structure of the membrane or structure of hemoglobin lead to a massive destruction of the RBCs, of which sickle cell anemia is important. An example of the parasitic infection which lead to anemia is the malaria. All of you know a malaria parasite is a protozoan. One single cell parasite invade the RBCs. Transmission occurs due to transmitted, the malaria parasite transmitted from patient to patient by the Anopheles mosquito, the female of Anopheles mosquito, and it will lead to excessive destruction of the RBCs because replication of the parasite will occur within the RBCs. It will lead to rupture of the RBCs, and then these parasites produce and will invade another cells. It will lead to massive destruction of the RBCs, and it lead to a type of anemia classified as hemolytic anemia. One of the other hemolytic anemias is the congenital anemia, of which the sickle cell anemia is important. This is a condition at which the RBCs will be converted into crescent shape when they are exposed to the side of the circulatory system which containing low oxygenation. And this conversion will occur due to a change in the beta chain of the sequences of amino acid. Actually, the amino acid 6, the glutamate will be replaced by valine. And this will lead to a different type of sequences. These sequences of amino acids will lead to formation of a new chain, beta chain. This will, and when the RBCs passing through, when the RBCs are hard, they are remaining disc-like concave as it is, with no change. But when they are passing through the circulatory system, at the areas at which the partial pressure of oxygen is decreased, the shape will be converted into crescent shape. And they will accumulate and will lead to blockage of the blood flow, in addition to their hemolysis. It is very serious type of congenital hemolytic anemia. Clear يا شباب؟ Clear يا شباب? Any question? Thank you.